Also, okay, awesome. Before I start with results and methods, I just want to make sure we're all on the same page. When I talk about dynamics in the context of physics, so not specifically applied to the brain, the dynamics of the system basically is defined as a quantifiable change over time in either the input or the output of said system. And in the case of the brain, it's in the output, right? Because we're interested in knowing how the brain varies in the response it generates. And in the context of, of the brain as an organism, dynamics are really relevant in that our brain is intrinsically variable and dynamic. And this is from a figure that Lucina loves, and it's all in her presentation of brain variability. And it goes, from the input or sensory input that it's that are coming in our brain to, the motor output that we're producing. And the important point of this figure or of this paper that has been made is the fact that what in the past the variability of the signal, whether it's of the microscale or the macroscale, was considered as a source of noise. But now we're more and more interested in the signal as a meaningful source to look at, because it has been shown to shape both brain function and structure. And so this is more or less the basis or the foundation of my whole PhD dissertation. Now, going back to physics, when we talk about dynamics, we can quantify this change over multiple timescales. So that's seconds, minutes, hours, years, and applying it at the spatial scale in the brain, we can look at things like neurons. So that's more at the microscale, or what we're all interested in, more macroscale neuroscience. We can look at regions, networks, or the famous Margulies functional hierarchy, separating, segregation, and integration. And in today's talk, I'll be talking about seconds and years, so in the context of aging, and looking at regions, networks, and years. Now, why do I care about brain dynamics? Well, the major headline of dynamics is that flexible brain dynamics are useful because they're useful for adaptive behavior. And we're all cognitive neuroscientists, and we want to look at behavior, right? And it has been shown recently that more static measures of brain function have less predictive power in terms of how they predict behavior compared to brain dynamics. And there has been over the years a lot of theoretical frameworks trying to define what brain dynamics mean. One idea is that this idea of stochastic facilitation. So the fact that the brain is intrinsically variable and dynamic is in a way helping neuronal function, because being variable or having relevant noise in the system enables effective neuronal function. Another hypothesis that has been put forward is the fact that the brain being dynamic basically enables the dynamicity of the brain enables the brain itself to produce probabilistic responses. And this is because the environment out there is highly dynamic as well. And so by exploring different possibility or having a greater dynamic range, what we can do is, or the brain is able to adapt to an ever-changing environment and not be static in the responses that it produces, but being able to successfully predict whatever is incoming. And this is a figure from Randy McIntosh 2010 paper, where he basically was showing a scenario in the absence of dynamics or variability. And you can see here, this system is pretty stagnant and that it's not really exploring its manifold. When we instead add variability or noise, what he calls, to the system, then the system itself is able to really explore its manifold or the dynamics. So these theoretical frameworks have been very helpful in trying to highlight that it's important to look at these brain dynamics. Now, in the context of how do we take these theories to actually methodological advances or experiments, that has been a bit of a tricky situation, especially with fMRI, because we know it's correlational. So I really think that animal studies and cellular and molecular studies have this power to really help us better understand the full picture. Okay, how do we quantify brain dynamics via fMRI? Well, you're probably all familiar. When I say dynamics, you're immediately going to think of dynamic connectivity. And this is something that has been studied over the years recently and has drawn a lot of attention. But less so when I talk about dynamics, less people think about bool variability. And this is because, variability and dynamic connectivity have been so far looked in silos as two separate concepts. And so in my work, what I try to do is really draw this link between these two separate concepts so far that I do think that are tied into one another. And the idea of, a link between variability and connectivity really comes from, as I said, seminal work from macroscale researchers. Look, basically, these groups have found that, neurons that tend to form synaptic connections with one another are the neurons that tend to also share cortical variability. And so this is, variability, for instance, in their spiking rate and so on and so forth. Now, in the context of, macroscale neuroscience, there have been, bits and pieces of work linking towards or hinting towards a link between variability and connectivity. But what I think, the work that I did with Jason, with Dina in 2021, I think, 2 years ago, last year, and Nathan has really been putting forward this idea that we should be looking at variability and connectivity as two intertwined concepts. I'm going to very quickly go over that paper because it's published and I want to go over minute results more in detail. So in that work, it's published in your image. What we did was we tried to, in a way, validate this idea that has been found at the microscale at the macroscale. So we had a data set of resting state fMRI, and we calculated static resting state connectivity. So it's the average, the correlation over time between pairs of regions. And then we related that to variability. Now, so far in the literature, the way variability has been computed or had been computed was at the level of single brain regions. So you get one metric per region. But because we wanted to draw a more close, relationship between variability and connectivity, I then, tried to identify a metric that allowed me to establish or quantify a measure of interregional variability that then I could relate to connectivity, because we know that each entry in a connectivity matrix indicates how similar the activity of two regions is. And so what I identified and defined was a metric of variability that basically quantified the similarity in variability levels between regions. And this, as I said, is near in the paper. Now, the main takeaway of that paper was that indeed interregional both signal variability traces connectivity, and that moment-to-moment changes in the both signal for each region is really related to the brain functional network architecture organization.
So, Julia, for the people who aren't always studying variability, which measure did you how did you quantify it?
Yes, absolutely. I have more slides later,
okay.
But so the way we define variability in this paper was using what's called the mean square successive difference. So we're talking about moment-to-moment changes in variability. So you'll see soon, it's basically the first order derivative of each regional both signal. So you're subtracting the signal from one time point to the next and looking at how it changes over time.
IMSSD means
high variability. Exactly. So the idea here was, okay, we established that these two concepts are linked, variability and connectivity. But in our near image paper, as I said, I was linking static connectivity to a dynamic measure, which is variability. And so in these subsequent, pieces of work that I've been working on was to really try and look at how connectivity dynamically unfolds over time and how this dynamic connectivity is related to dynamic or the dynamics of each region, so both variability. And this is basically one of the studies I'll be talking about today, where, as I said, I'm trying to draw this link between regional dynamics and network dynamics. But before we go into that, I just want to take a step back and put on my, my methods hat and talk about how I felt when I approached the field of variability. And this is, I don't know if you're familiar with Dante Alighieri, the Divine Comedy. And this is basically me trying to navigate the brain variability literature. This is the incipit, so the start of the book that he wrote. And it's basically, in summary, him finding himself in the midst of a very, very wild forest. So he's he's not able to figure out where to go. He can't see anything, but he knows that, there's something out there, and he's thriving and like trying to figure out how to get there. So it's basically me. As soon as I was talking to Nathan, I was just like, okay, I want, to study variability, but I really don't know where to go. I can't see anything right now. It's so dark. And so in my PhD, or over these years of my PhD, while trying to, from a cognitive neuroscience perspective, trying to make advances in, how brain variability is helpful in the context of psychology and cognitive neuroscience, I also figured out that for my own sanity and clarity, I also wanted to put on my methods hat and trying to understand, what variability actually means for a more, theoretical perspective. And so I'll start actually from my third study, where what I'm doing is I'm really trying to do some work that is very boring and tedious, such as trying different combinations of different types of acquisition, pre-processing, metrics, and so on and so forth, to try and see what all of these steps in, the pipeline that we have to, you do to get to a bold variability metric, actually what's the impact of all of those. And this is part of a review that I'm writing, where not only I am, running your standard meta-analysis of, all the papers that have looked at variability throughout the years, but specifically, I really hope to shed some light on, bold variability and try and guide the field of, people that are approaching, or the word the little Dantes, me, a few years ago, because I really believe, and I think that it may be helpful, not just for me, but for everyone else. So let's start from this part, because I do think that it's really helpful to know these things before even we start interpreting my findings of study 2.
And this is important because you haven't mentioned that there's not very many labs working on variability. So there's like Doug Garrett, who's basically the only person in the world in Germany who has done a lot of this work. But I don't know, maybe Bratislav's group, but there's not that many groups coming up with these methods and applying them and really thinking about it. So everything you do is like you're doing it for the first time.
Yeah, that's it. That's also why I was so confused. Okay, so by now you probably know that I love analogies because they help me create clarity. So I spent so much time trying to figure out, how do I clearly convey the idea that this is a mess? And, we should. Yeah, so this is actually something I drew with my undergrad, and this is going to be the graphical abstract of the review. So I'm a terrible gardener, so I don't know why I came up with it, but I promise it makes sense. So the idea here is that really studying both signal variability is similar to growing tomatoes from seeds. And this is, from acquiring or buying your own tomatoes or having them, in your backyard, to actually having your final product, which is the tomato itself. And in the context of bold variability, basically the idea here is that at every step, so every single step of this chain, from how you acquire your data, whether it's open data or your own data, and the 3 million different pipelines or acquisition parameters you can have, to how you pre-process your data, how you clean the soil, how you cut the grass that you don't want, to how you normalize a time series, or in the context of tomatoes, whether you're putting them in different tomato strains, and how you derive your variability metric. Are you looking at the growth of the whole plant? So that's we're going to talk about standard deviation. Or are you interested in the single tomatoes? And that will be more of the MSSD. All of these steps actually do matter because they do impact your final result. And this is important because, as Lucina said, not that many people do variability work. And so when somebody reads a variability paper, they just, draw general inferences or general conclusions. But at the end of the day, it's really because there's not that many people out there in variability, and B, people are not aware of all of these implications. We can't really, as of now, generalize beyond that study, unfortunately. Okay, so let's start. For the sake of time, I'm not going to go into details over all of the steps. I'm going to talk more extensively about pre-processing and metrics. But I'll talk about data acquisition because this is a huge You know, something that has taken a lot of time. And thanks to, you know, Tina and Jason for giving me access to the NKI data set, and we'll talk about it in a second.
Well, Jason did something, right, like similar, you know, you presented this at OHPM, was that 2 years ago, or where was
Oh,
it?
no, that was just test requests.
That was test reliability of MSSD,
Yeah.
right, and that's probably, that meeting was like 2 or 3 years ago.
I was in Rome.
Oh, it was Rome, okay. So
2019,
2019.
yeah.
So that's when we probably first started talking with you guys,
Yeah,
because
With Nathan.
yeah,
Nathan and
exactly.
and everyone.
exactly.
So basically, I mean, all of these methods papers are kind of necessary when you're wading
That's
into,
it.
like new territory. It's like you don't want to write a method.
That's it.
It's like, no, nobody wants to. But then you like have to in order to like establish what you're doing.
Exactly. So that's what I'm trying to do. But yeah. So this has been so boring and tedious, but here we go. So here I have 4 different data sets that have completely different acquisition protocols. And the goal here is just to investigate the impact of, you know, all these different acquisition parameters on full signal variability. And I apologize, this is a busy slide, but we have 4 different data sets. So we have the Cornell data that is a multi-echo single band data set. Then we have PreventAD, which is multi-echo multi-band. And then we have NKI, which is single echo multi-band, and Toronto, which is single. So single echo, single band. So we have all possible combinations. And I've been having a headache because I need to pre-process all of these. But I'm getting there. I'm almost there. Anyways, so I'm not going to show you results on this because I'm finalizing the last. I'm doing ICA fix. I don't know if you're familiar with it, but you have to manually say what's noise and signal.
Yeah.
And I'm going over like 5 million different brains.
I think we did ICA fix on it.
So I don't have to trust that. Yeah, unfortunately, like fortunately, not that.
That's the one that I was talking. Did you?
A lot, yeah. Yes.
You can thank them.
Thank you guys. Hearing me with one data set. But anyways, so stay tuned for these results, but I think it will be very useful. And you'll see as I go over my results later that it does acquisition does impact our variability metric. But let's switch gears and talk about pre-processing. So here what I used was single echo data and not multi-echo. Multi-echo is fairly new, and I believe our neuroimage paper is the only one that is used in multi-echo and variability. So I thought, you know, if I'm writing a guideline, like guidelines for the field, it makes no sense to use multi-echo. So this is why I use single echo, and it's an adult lifespan data set. And here, yes.
Sorry, can you just
Clarify? Yes.
Or just define multi-echo?
Yes. So typically, you know, when you collect fMRI data, you send, you know, one pulse or one echo, and that's single echo data. But multi-echo means that in the same amount of time, more or less, you're sending 3 different echoes. So you're collecting technically more data over the same amount of time, more or less. And the idea is that you're maximizing, you know, spatial resolution, because different parts of the brain have signal decay in their signal at different times. So if you send, let's say, an echo at 15, 30, and 45 seconds, then you're able to have a better spatial coverage of the full brain. So for instance, areas, you know, near the sinuses or orbital frontal cortex typically have a lot of signal dropout in single echo fMRI. But multi-echo, because, you know, we're reading out signals at multiple times, we can better delineate the signal in those regions. However, thanks for this question. The problem here, though, of multi-echo, this is maybe one thing to keep in mind, is that the TR is way slower because, you know, TE and TR. So when you read out the signal and when you send the signal are interdependent. So if you are slowing down or increasing your TE, then you're slowing down your TR. So typically in a fMRI study, the TR is of 2 seconds. But in multi-echo, because we're reading out signal on multiple time points, then our TR is slower. That means that if you're doing dynamics, that's like one of the comments that we got, it may not be ideal because we're just slower. However, our results are more robust because we have more data. So this is kind of like, you know, a drill back.
Trade-off.
Exactly, that you need to take
But
into
you
account.
have more data, so you have.
You have more readouts.
More readouts, yeah.
So if a TR is 2, means you collect a whole brain image every 2 seconds, right? But in a normal, not single echo, at that every 2 seconds, you get the one readout, right?
Correct.
She's talking about more than one readout every 2 seconds.
Yeah.
If it's multi-echo means 1, 2 or more, or.
Yeah, two or
so-
more.
So what data you're working?
Here we have 3.
3, okay, so you have like, so you can't fit out 3 readouts in a TR of 2, so you have to extend the TR. What is it then? The TR is 3?
3. Yeah.
Okay, so that's once every 3 seconds you're getting whole brain image, which is obviously slower. And if you know what HCP and some of the data sets will work, those are TR of 7. So that doesn't have multi-echo, right? That's just something,
Exactly.
but it's got data every 0.7, which is great for dynamics because you're sampling it. So this seems like an interesting trade-off that I hadn't thought of before. That
Mhm.
certainly you have to make these decisions.
That's it. That's it.
When you get 3 readouts, they get reduced back to 1 on the TR.
Okay, so
At
great
that
question.
point.
So then,
Yeah, I don't understand.
Yeah.
why you have to have the same subject.
Yeah, I don't have something in this.
so it all comes down to how you can process your data. So when you have multi-echo, what you do is you do PCA. And then what you do is you optionally combine all these 3 echoes, all these 3 readouts to 1. So at the end of the day, it's like single echo.
It ends up looking the same,
Exactly.
But the data, because it's
collected.
collected, because there's 3 and you're sort of reducing it, it's smart.
That's it. That's when I say we have more data, right? That's what I meant.
Well, you've
Okay.
been. you-
Better
you
To start off
Data,
with.
better data quality.
Exactly.
have to
Exactly.
separate the noise better. right?
Exactly.
And then I guess that matters how you reduce it to 1.
Yes.
Okay.
Yes.
This is relevant. What we were talking about yesterday with the little signal.
Yeah, because we don't do global signal
Yeah.
regression, right? Yeah.
Okay, so
Exactly.
that's.
Okay.
Good to
So
Well,
yes.
and then also the TR multi. So some people say that multi-band is only good for removing noise, just because the whole signal is so sluggish. So that's another response.
Mhm.
But whole signal is typically 0.01 to 0.15.
Yeah, exactly.
So 0.7 TR
Doesn't
compared to
help
0.2 doesn't really affect the whole signal. It's just gonna allow you to remove more noise.
More noise. Yep.
So-
Exactly. I think the future, this is one data set that we have is multi-echo multi-band. Because here we have a TR of 1, I think. So it's very fast,
Oh, yeah.
but it's also multi-echo.
Yeah.
Unfortunately, though, this is prevented. So this is a data set that is preclinical Alzheimer's. So it's not your healthy standard subjects. So I can't write a paper just on that, on variability. But
10 subjects.
no, this is, we have 181, but because I didn't want to do ICA fix on 100.
Wait, I say fixed doesn't matter though, once you turn it on 20.
Well, true, but I didn't want to do more than 10. I was just, because I have so many data sets. And here it's
Wait,
just
so you
like a
trained
proof of
10
concept.
subjects in there?
Sorry,
You trained 10 subjects?
this is, I have, yeah, exactly. I
You
have
trained
like
10.
yeah.
You can just run it on all the rest.
just on 10. Don't you have like
You
a
don't have to do every single subject individually, right?
Yeah, no, I see a fixed means you're training the classifier. So typically you do 16. Well, that's what they recommend, but you can
Okay.
get results with 10.
Okay, so maybe I should look into
You
that.
can run the
Clean
whole
all the
data
rest.
set
Yeah.
from the 10. Yeah.
Okay.
I mean, based on the 10.
Yeah.
Yeah.
So make a training file, classify a novel subject and see how it classifies. And then you
Okay.
can use the whole data set.
Yeah, you don't have to do much more once you've already done.
Yeah, once you classify the 16, then everything else should just be
Awesome.
processed. So, yeah, that's what
Okay.
you can do.
Okay, I will. Okay, so for pre-processing, what I did was I investigated the impact of 6 different pre-processing strategies on both variability. So here, as I said, we have NBAC. And to determine how the pre-processing pipelines actually affect both variability, I looked at how it impacted the results in terms of effects of working memory, load, age, and load by age interaction. So these are the different pipelines. The important part is whether we did ICA fix or aroma, and whether we added, the covariant regression. So whether we were covariant out, motion, white matter, CSF, and vessel. So very quickly, I'm going to go over main effects of ST, of load, age, and the interaction. So here, the main point, if you want more results about, the effects per se, you can look at our paper. But the idea here is that what I found was that in general, each pipeline was sensitive to the effects of load on bold SD, and that there was a decrease as a function of load. So the greater the load, the decrease in bold variability. But in terms of which pipeline was the best in estimating bold variability, we found that the ones without the noising had somewhat like smaller extents of the effect. And so what we say here is that, regressing out additional covariance actually helps when, your effect sizes are pretty small if you're looking at individual differences, or in this case, age, because it's the same idea here. Basically, additional covariant regression seems necessary for any pipeline to see at least some minimal age effects, as you can see in these brains. And it's pretty much the same when we're looking at the interaction. So taken together, my conclusion of this part of the review was that aggressive noise removal on single echo data during pre-processing may have more of an influence when the effect size, as I said, is smaller or related to individual differences in which noise is a known confound, as we know in aging populations. So, and this is something that also Doug Garrett has been, applying in his own work, but nobody has ever actually looked at whether this is the case or not. So now the fun part. Sorry?
Didn't he look at that in his first paper?
I don't know. Did he? In aging? Okay, I
Yeah,
don't
I'm
know.
pretty sure he showed that with and without.
Okay.
I see denoising,
Okay,
the effect size double.
but it wasn't was also the covariant regression?
Probably not all these.
No, it's just I see denoising versus not.
Yeah,
And I
okay.
see denoising increases the effect size.
That's it. That's what we're seeing as
Yeah.
well. But, extensively across all six. Okay. Now something that is very interesting, because this basically opened a can of worms for me. So here what I wanted to do was to look at the impact of variability metric and deciding whether to use SD or MSSD impacts our results. Because true that throughout in my own paper there was a high correlation of 0.9 between SD and MSSD. And also in Doug's work, and I believe also in yours, Jason. The problem here is that I do think that these things capture different things or different aspects of the time series. And I hope that I'll convince you through these simulations that I ran and some real fMRI data.
Well, you can imagine a scenario in which the SD is, let's see, the MSSD is low, but the SD is. Right?
Yeah, I'll show you.
Yeah, okay.
Yeah. So, well, first, okay, so just to make sure that we're all on the same page, what am I talking about? So when the field of fold variability actually started with Doug Garrett, what he was talking about was standard deviation. So what he was doing, he was just looking at the overall temporal deviation in each regional fold signal. So he was just taking the standard deviation in each region. But then, thanks to work also from Jason and Lucina, the field also has started to consider MSSD as a meaningful source of fold variability. And this is what I said before, the first order derivative of each regional time series. So you're looking at how the signal is changing from one time point to the next. Now, from a mathematical standpoint, you could think that these two metrics actually do capture different parts of the time series. And so to bring or drive home this point, what I did was I ran some very basic simulations. When people say, you need something very simple to drive home the point. And this is, so it's like, I don't know, grade 1 math. So what I did here, I created two scenarios where I was manipulating amplitude and holding frequency constant, and then manipulating frequency and holding amplitude constant. So this is scenario number 1. What I did was I simply, increased amplitude of a regular sine wave going from the top to the bottom. And as you can see, both metrics, so MSSD and standard deviation, are increasing. Now, the interesting thing is that if you keep amplitude constant and you manipulate the frequency, what's happening, as expected from the mathematical formula of the two, MSSD is impacted, but standard deviation is not. And so as you go from the top to the bottom, what you see is that MSSD is decreasing because, as you can see, there's less frequency. So the signal is getting slower, but standard deviation is not showing any change. And so this is, in a way, very interesting because it's driving home the point that these two metrics, sure, they may be related to one another, but they're actually capturing different things, different aspects of the time series. And this is basically what you were saying before, Lucina. So here is, instead of using toy examples, I picked, some real fMRI data. And here I'm showing two regions that have, in green, we have a region that has high standard deviation because, as you can see, the amplitude is bigger. but the MSSD is relatively low. And in orange, we have a time series that is more compact, but it has high frequency. So the MSSD is higher, but because the amplitude is lower, then we have a lower SC. And so when I first showed these results to Nathan, he was like, okay, are you saying that MSSD is better? I don't want to create any enemies, but I do think that's the case. And there were
But that's
three
what I
reasons.
can. So how we decide 5 years. ago
Yes,
to
yes.
Do MSSD rather than ST.
Well, you'll see soon as well, but I think that SC is too coarse.
Yeah.
It's too coarse of a measure, and it's really dependent on any shift that may happen to the data. So if you have a mean shift, SC will be impacted, but MSSD will not. And so my conclusion in this work is that MSSD is, frequency dependent, as we saw, and this moment-to-moment speed of change, and standard deviation is more related to amplitude, so it's this magnitude of change. And because I'm interested in MSSD, what these results tell us is that this metric heavily depends on how, so the frequency range and where, so the time points that you include in the time series you're sampling. And so here the guidelines that I'm offering to the field is not so much go with MSSD, even though I wish I could say that. But you do have to pick a metric and know what your metric is telling you, and then interpret your findings based on that. And then potentially do try and generalize your findings with the other one, and interpret, as I said, what you obtain accordingly. And now something very interesting that I found, and this is a liaison to my second study. So because I'm putting on back my methods hat, I wanted to see whether the last bit of our tomato chain, so the variability metric, is impacted by our first step. So that's the data acquisition, right? And so what I did was using our slowest data set, so Cornell, which is, as we said, a multi-echo data set, and our fastest data set, so that's our NKI, right? So we have single echo, but it's multi-band. What happens when we derive MSSD and SC in these two data sets, and then we relate these metrics with one another? Not surprisingly, what we found was that if we look at standard deviation, these two metrics are related in the two data sets. So we have Cornell on the bottom and on the x-axis, and NPI on the y-axis. And this is not surprising because SC is pretty coarse, as we said, and as we saw, it's related to the amplitude of change. And so instead, when you look at MSSD, these two metrics are not related between the two data sets. And so this, in a way, at first kind of scared me, because then I thought, okay, does this mean that we can't really generalize any finding in the variability literature because it all depends on how you collect your data? But then now I'm basically going down this rabbit hole when I'm trying to test all possible combinations and see whether it's the case, or whether there is something more than just like scanner dependency in MSSD metrics. But it's definitely something that I think the field should know, and we should keep in mind when we look at variability metric. Okay.
The TR is different between both?
Yes, because this is 3 and this is 1, right? So that's my point is, or what I'm thinking is that because MSSD is looking at really data points versus SC is just one average, of course, metric over the whole time series, it makes sense that MSSD is highly dependent on how you collect your data, because we're looking at, the nitty-gritty of, time points by time point.
Yeah, well, because just the
Exactly.
longer the TR is, the farther the signal from.
That's it.
So
Mhm.
I think you would need to get TRs the same to do that comparison.
That's exactly what I'm doing. So what I'm doing right now, I'm making Cornell faster and NPI slower, and then creating 3 million things.
Well,
But
NKI has 2 different TRs. That's what Jason was using, right?
Oh, more fast.
I know one was fast.
0.7.
One was
Oh, wait,
1.5.
2.5.
One was 1.4 and one was 0.7.
0.64.
Yeah.
But they have also a 2.
So you have 3
They have 3
TRs in
TRs,
NKI.
I think. I'm pretty sure,
Well, you used 2 of them in your paper.
the 2 fastest.
Yeah.
But I think
Okay.
there was a slower 2.5, and that will get you closer to.
To Cornell. Okay, I'll
Because
look
are you
into
trying to do it to downsampling and upsampling with yours?
That's it.
Because you're essentially filtering it to that.
Mhm.
So. Yeah. that's
I know.
tough.
I don't know why I'm going down this rabbit hole, but here we are.
Yeah.
Okay, I'll look into that. Okay, Now, because I want to talk to you about what I'm most excited about, which is our study, the third study. What I talked about so far is just boring, but I think it's useful. Let's now talk about more exciting things.
Depends on your preference.
Yeah, exactly.
No, this is more exciting. Be ready, guys.
Be ready.
Popcorn science.
Or is someone to do that other stuff.
Well, Jason, you're on that paper, so if you want to help me out. Okay, here we go. So now we're basically going back to the second study where I'm trying to link dynamic connectivity with variability and a bunch of other things. So again, before we go into the aims of that study, I want to show you why it's important to link connectivity with variability. And here, because I'm very close to the data, I want to show you an example of why these two concepts are intertwined. And this is some work that I did with my undergrad at McGill. So what we did here, I wanted to ask the question, how do initial or regional MSSD levels impact connectivity from a static point of view? Because if I can show that two regions that have a similar MSSD level from a more closer to the data, then if that's the case, that two regions have a similar MSSD level, then is it the case that these levels of MSSD are actually linked to a higher connectivity? And so what I did here on the left, focus on these histograms on the left, we plotted the distribution of MSSD across regions, so for each region, and this is a really normal distribution, and then we binned regions with low, medium, and high MSSD. And then we picked pairs. So we selected pairs, all possible combinations of pairs with low and medium, medium and high, low and high. And we looked at how initial MSSD impacts the connectivity of those two brain regions. And this is basically validating once again our near image paper, where we're showing that two regions that have similar initial MSSD levels, so they're varying similarly over time, actually have high connectivity. So this is a scenario where we have low and low MSSD, two regions. And as you can see, you have a high connectivity of 0.9. And this is a scenario where we have distinct or opposite levels of variability. This is a region with low and a region with high, and the connectivity is low, so it's 0.24. So again, this is a way to set the foundation, guys. We should look at these things as one. So
Are you determining the data?
no, because it's multi-echo.
You can't determine.
There's no D-trend on multi-echo because it's all biophysical models and so on and so forth. There's no D-trend.
Because one of the issues or the differences between MSSD and SD is that MSSD accounts for the return.
Okay,
So that's
so you think that SD is
there's a methods paper like Journal of Neuroscience Methods
okay?
or a comment or something on Doug Garrett's paper where they say, oh, should use MSSD because it's not susceptible to trends.
MSSD.
Yeah, so they show it like a little simulation. Doug does something because he goes across trials or something to account for it. So the trend is
Okay, but that's good. If we're using MSSD, it doesn't matter, right?
yes.
Okay,
But then that
perfect.
could be causing the SD to change so between these.
Okay.
So
Yeah. But my point here, well, I added SD, but here I'm just looking at MSSD. So
okay.
yeah. No, like good point.
But you're comparing it to
Yeah,
SD, the
yeah,
trend is gonna matter.
That's a great point. Okay. So what are the questions that I wanted to ask in this study? So first, because full variability literature typically is just relating variability to something else. So there hasn't been in my, as far as I know, papers looking at how variability is spatially organized. And I think this is important because if we establish that some regions to start with are more variable than others, then it's more likely that whenever we correlate variability with anything else, then there's a higher correlation if your initial levels of variability are higher, versus if your range of variability is lower, then it makes more sense that that region may show a lower correlation with something
Why
else.
hasn't there been like a paper just basically looking at across the brain?
There has been?
Why hasn't there?
I don't know.
That seems like the first question. Wait. No, Doug hasn't done any of that?
I don't think, because it's always like variability related to age or
and-
cognition
Right.
or,
Yeah, I'm surprised there.
Michael Cole does some stuff where he looks at how does variability or what he calls input noise influence connectivity.
Okay,
Like the poll as a journal of neuroscience. That's or something. But
okay,
basically they show that, well, this is why I think you can argue that variability is also interesting. Because it's not affected by these. Basically, when you look at FC,
mhm,
the two sources of noise from each node can influence the connectivity value, right? So you can increase or decrease whatever. But for one node, when you're just looking at variability, I think you can average that out maybe, whereas you can't do that. I don't know.
okay,
I was trying to formulate this argument more defense, but
Okay.
yeah.
okay, I'll look at that paper.
But yeah, no, there's a lot of modeling work looking at how do sources of noise or variability on one node or two nodes influence the connectivity.
Mhm.
Great. So you're telling me there's no paper on just like the topography of variability across the cortex?
No, because I couldn't find it.
Huh.
So that's why I'm like, okay, let's establish this first before we do anything else.
Yeah.
You know, but I may be wrong.
I can't get one.
Yeah, so that's, I started the paper talking about that. And then what I wanted to do, and this is something that has been really exciting, I wanted to define a new metric of dynamic connectivity that in a way was minimizing any user input. And this is because when you think about connectivity, what typically people do is, they window the time series and then they have to cluster and select how many clusters and so on and so forth. And there has been shown that, it depends, or results may be dependent on these, different decision points that researchers have to make or to do. And lastly, so I wanted them to relate connectivity, so dynamic connectivity with full variability, and define what it means for a region to be dynamic in the context of graph theory. Graph theory has been very influential in the context of connectomics because it really helps us delineate the topography of brain network organization. And now the fun part was to try and basically bring together a bunch of silos paper that have been looking at the importance of dynamics or the relationship between dynamics and a bunch of other things like the spatial temporal properties of the both signals, such as temporal autocorrelation, but also more hardwired things about the brain. So site architecture, microstructure, cortical evolution, and neuromodulation. If you think about Max Schein's work or Michael Brexpier, and try to really bring all of these things in one model and make them compete against one another. What is actually shaping brain dynamics? And lastly, because I'm a cognitive neuroscientist, I wanted to look at the impact of age and behavior and cognition. This is a lot. I'll try to be quick and talk about all of these things. And then I put on, again, my methods hat, and I wanted to validate all of these things on the NKI data and the Toronto data. So this is a huge paper, and I'm regretting the decision of putting all of this in one paper. But
Make it into two.
I could, but Nathan now is like, nature. We're doing this. It's not. It could be my thesis. But this is the paper that you guys are on too. So here we go. Okay, so let's start by characterizing brain maps of variability. So the exciting result that we got here is that variability levels, not so surprising, do vary in a meaningful way, in a spatially meaningful way. And if you're familiar a little bit with the control network, for instance, that I can make my point here, this is a region that is the middle frontal gyrus. And when I showed these results to Nathan, he was like, basically, okay, this is nature ready, because Nathan basically taught me, which I didn't know, that the middle frontal gyrus is a very dynamic region in terms of its role in cognitive control and executive function. And what I'm showing here indeed is that in fitting it fits the idea that it's the dynamic region, and that among all these other gyrus in the same gyrus, so the anterior portion or the posterior portion, the middle frontal gyrus is the one that is showing the highest variability. And so there may be, a link between the dynamics of the system, in this case of this region, and things like, flexible behavior. And this is something that we're all interested in. Anyways, it may be easier to look at the dynamics on this bar plot. So here what I'm doing is I'm collapsing all regions based on their network assignment. and you're going from, visual systems to control and default. So from segregation to, integration. And the point here that I want to make is that variability levels do tend to change or vary along this axis, and that regions that are more segregated, such as, the visual system, tend to have a higher variability versus regions that are more, feeding or higher order networks, such as the control and the default, tend to be more, or integrated information tend to be more stable. And this, when I looked at this result, I thought, okay, this makes a lot of sense, because the control and the default have to, integrate information coming from different sources. So it makes sense that they're more stable. In a way, they're more predictable, in that their signal tends to be more similar over time, so that other regions can predict what signal they may, get from these two networks.
I would've
However,
thought the opposite.
wait for it. However, I did the exact same on the NKI, and we have a flip here on the
Is
axis.
it eyes open or eyes closed?
This is eye closed. Eye closed. And then
Eyes
in the
closed.
NKI, no, wait, is it eyes closed? No, I think it's eyes open, because
Eyes open?
they're staring
Yeah,
at the
that's right.
cross.
Oh, that's
Yeah,
the
that's
visual
what-
network.
Yeah, eyes open.
Yeah.
Okay. But yeah, when you do,
We just do an eyes open, eyes closed comparison, right?
I don't know what NKI is. It's NKI.
It's probably eyes open, but.
I think it's open,
So, okay.
but I don't know. But yeah, when I calculated this on the NKI, it was the opposite. So you had higher order systems that had higher variability compared to lower level system. But this is again going back to the idea that MSST is not replicating between the two data sets. So I paused on this, and I need to dig deeper into this. But okay, so how did I define dynamic connectivity? Because this is one of the big exciting new things of this paper. So here, what I did was I thought back on my time in Toronto, and this is when Hervé Abdi came to give a talk. I don't know if you guys are familiar. He's a statistician at UT Dallas, and he's a French dude, as you can see here in my example with wine. But anyways, so what he did was he came to give a lecture on PCA and multidimensional scaling, and he was using this method called costatus, which is similar or like a fancy PCA version. And he used it to explain to us what PCA and MDS are. And he was using examples like wine, cheese, Asian noodles. And then my time in Nathan Black came, and I thought, wow, if I could use that somehow in my PhD, that would be great. So here we are. So I'm using costatus, and I'll explain to you now in a second with another analogy, to establish dynamic connectivity. So the way it works is, let's say we are trying to establish, different, how different wines are similar to one another. So we have 12 different wines and 10 different experts. And our goal is to characterize how similar these wines are and find out if there's any agreement between the 10 different assessors that are asked to evaluate these wines. If we do a simple PCA, and this is how costatus starts, you basically get, in this PC1, PC2 space, which is called compromise space, across the expert a score for each wine, right? And so wines that are closer together are rated more similarly across experts, and wines that are further apart from each other, then they're more rated more dissimilarly. Now, what costatus allows you to do is for each assessor to project back basically the rating for each wine. And more specifically, what we're able to do is back to our average compromise space. So we have in the middle, this is the average ratings across all assessors for each wine. What you can do is around each average, plot each assessor rating. So each dot is one assessor. And the idea is that the further apart, or the further the assessor is from the rating mean, the more dissimilar it is from, all other assessors. Now, keep this in mind as we go back to our brain. So what I did here, I, basically windowed all time series using, 45-second window, which is the gold standard in the field. And then I applied costatus to try to find one metric of dynamic connectivity that was entirely data driven, where I did not have to, to decide how many cluster I wanted and so on and so forth. And so what this looks like, after windowing the time series, the idea is to bring together all the windows for all subjects and look at how similar these windows are over time, and obtain, similarly to our wines, a compromise space, where you have, it's an optimum average of all the windows of all people over time. So you're gonna give, a higher or a bigger weight to those connections that are more represented over windows and over people. Then what you can do is you do a PCA or SVD on this compromised space, and you back project, just like we did it with our Y, for each region, their connectivity patterns over time for each subject. So more specifically, what you can get it here, and this is the goal of using code status in dynamic connectivity. For each region, you define how flexible or stable it is over time for each subject. So we have the mean connectivity for each region in the center, and then each dot here, each purple dot, is one window. So it's the connectivity of that region for that window. And so the idea here is that mathematically, what you can do is you can draw the area of the hall around the center. So the more flexible a region is, again, based on Euclidean distance, the bigger the area is, right? Because that means that its connectivity patterns over time are more diverse because they diverge more from the center. So that's the mean, right, for each subject. Instead, if the area of the hall is smaller, that means that that region has a more stable connectivity profile because all the dots then are more clustered together. That means that they're more similar to one another in Euclidean distance term. And so I was really happy applying this code status to connectivity because this really allows us to do basic statistics because now we just have one metric, right, for each region, for each subject, and we can just look at or relate, the flexibility or stability of each region to a bunch of other things. And again, we wanted to delineate, the dynamics. So having the area of the hall, this is how I refer to each regional metric because it's technically the area of the hall, right? Then you can just look at the topography of the dynamic connectivity, which strikingly is very similar to variability. So this is, I'm showing, dynamic connectivity, and then this other pattern is variability. And as you can see, across these networks, these two things relate. And very nicely, when you look at, two different data sets, so you're looking within data set. I'm not, relating metrics across data sets. So I'm relating variability and area of the hall within our main data set. So this is Cornell, and then this is for NKI. We're really driving home the point that regional dynamics do relate to network dynamics because area of the hall is relating to MSSD, both in Cornell and in NKI. Then very quickly, we wanted to define what it means for a region to be dynamic or like flexible or stable. And so I related this to a bunch of, graph theory metrics that define integration and segregation. For purpose of time, I'm just going to focus on results of participation coefficient. And before I go into the results, high levels of participation coefficient indicate higher integration. So this means it's a region that is talking to a lot of other regions. And so here what we found, apologize, I need to figure out how to best visualize this because this is a PLS result and I'm not going to spend time going over that. But the idea here is that what we found is that more or less homogeneously across the brain, what we found was that a smaller area of the hall, so a region that is more stable or has more stable dynamics, is linked with greater participation coefficient. And so this means that there is greater integration. So basically it feeds back to our Cornell results where we show that higher order networks such as the control and default are more stable because those are the ones that are integrating more information across the brain. And so in this set, I define a new metric of network brain dynamics that is robust, reliable, and less arbitrary. And I show that regional signal variability follows a precise spatial topography that then is recapitulated at the level of network brain dynamics, and that stable regional dynamics are associated with greater integration. Now, in the last minutes of this talk, I want to basically talk to you about this new venue that I opened looking at dynamics across, or the covariance of dynamics with a bunch of other things. This bunch of other things is, properties of the time series, functional hierarchy, cortical evolution, cell architecture and microstructure, metabolism, and neuromodulation. And here I linked some, citations or resources that motivated my question. So I'm not going to go too much in the details of that. But what I really came in handy was two toolboxes that I've been using to relate dynamics to these things. And one is Neuromaps, which is an effort led by Bratislav Misic and McGill. So this is a toolbox that includes maps, so spatial maps of the things that are highlighted in green. And then the Big Brain Warp, which is a singular data set that is basically providing us with high resolution things, cyto and microstructure from a postmortem human brain that was stained with the Merker technique. So here, what you really can do is, parcellate all of these maps in whatever parcellation you want. In my case, it was Schaefer 217, and then you get a regional estimate of all of these things. And then because we have regional estimates of dynamics now, because we have our area of the Hall and we have variability, then we can create a model where we're trying to find the covariance of all of these things. And you can answer questions like, how are all of these things relating to regional and network dynamics? Or you can see how all of these factors compete against each other to predict dynamics at the regional and brain at the network level.
This is good if you go back to that slide, this one, yeah, I didn't know, I had heard about the brain maps paper, but I didn't know what it was because I haven't read it yet, but this is going to be relevant for Priyanka because she's just working on trying to link, like genetics and metabolics and things like that with all of these brain measures in autism. I didn't know about this thing, but I think that's something we should look at.
You should look at that.
This one right here, Neuromaps, because that's I saw it when it came out on Twitter and I never read it, but it's like it looks like it's going to be exactly what some of what you're proposing for the grants.
I do one thing to keep in mind is for instance here I could do that because I have a young adult sample, but I could never relate this to my aging sample just because you need to look at the ages of where these maps are from. And some of these things have a relatively small sample. So you have to I did some cherry picking in terms of what maps I wanted just because you want robust, results. But I'm happy I have a whole spreadsheet of, all these details. So I'm happy to help you.
Yeah, I should.
Yeah, perfect.
Thank you.
No worries. Okay, so quickly, because it's a lot, I'm just going to go over site architecture, microstructure, and receptors. It's something that people haven't, as I said, in silos looked at in relation to dynamics, but not really together. So this is cyto and microstructure. I'm not at all versed in this field, so I got the help from my undergrad, she's doing a rotation in Michael Fertrides' lab. I don't know if you guys are familiar with him, but he's a big person and
person.
Exactly.
Yeah, we did a bunch of stuff with him. I was a postdoc, and he's like this great anatomist / physiologist where he painstakingly injects the monkey brain to figure out, where the axons go, and then he also trains the monkey on different tasks and does, and he does it himself. He's like this old man who's still doing all this stuff on his own. Why don't you just relax in your office? But he was really into it.
Yeah, still is.
It's amazing.
Still is.
I'm never gonna be like that. But I'm already someone else do this.
Yeah.
It's hard work.
No, absolutely. Yeah, so I'm still interpreting these results, but you'll see in a second why I'm talking about it now. So bear with me. So here I'm relating the area of the Hall, so our network dynamics to cyto and microstructure. So we have thickness of layers 1 and 6. We have this histological gradient that tells us the site architectural differentiation. So here is differences in cell morphology, cell density, thickness, and transition between the different layers. And then you have a microstructural gradient. This is derived from an in vivo MRI. So this is the T1 versus T2 ratio. So it's basically myelin content. And again, higher scores in these gradients indicate higher differentiation. Now, when you relate these things to dynamics, here I am not entirely sure what the interpretation is, but there is a relationship and I'm working on interpreting this. The idea here, what I found is that high dynamics, so regions that have high dynamics or are more flexible, have higher scores or higher differentiation on the first histological gradient and layer 4. And this is very interesting because layer 4, as I mentioned here, is the layer that is defining external input. And I believe that it's one of the layers that is not really or highly expressed in higher order cortex. And it kind of like ties back to the idea that,
Layer 4 was the input. Layer,
isn't it 1,
elements.
2, 3?
I have to revisit.
Yeah, me too. I need to like look into that as I'm interpreting the finding. But I promise there's meaning in this. I just have to like wrap my head around it. But we found something. And the very interesting thing is that there is validation across data sets. So my point here is that true that dynamics are highly dependent on, how you acquire your data, and we've seen this. But as soon as you relate dynamics to something else that is not relating to time series, then those results do replicate, because this is Cornell and this is NKI. And as you can see, so whatever is not crossing 0 is significant. These effects are pointing in the same direction. Sure, there's not a full replication, but I'm not expecting a full replication, but effects are there and they hold. Now, what I did interpret more is the relationship between area of the Hall and receptor densities. And I'm very excited about these results because in the variability literature, that kind of started paving the path towards saying that dopamine and GABA are relevant to receptor densities, or to, full variability. But so far, no one has looked at a full characterization of, receptors and variability, or receptors and dynamics. And here we have a variety of different receptors spanning, more inhibition and excitatory type of receptors. And what we found, again, I want to highlight, and I'm so proud of this result, that there's a validation in Cornell and NKI, despite the highly differences in, dynamics between these two data sets. And they all, sure, it's not fully, again, replication, but are pointing toward the same direction. And what I found here is that regions that have higher dynamics have higher expression of not just dopamine and GABA, and this is great because we're replicating DUG, but also glutamate and serotonin. And again, I'm trying to interpret these in more detail, but I find that these are very exciting results for the field, and they may, guide future investigations.
Yeah, this could be a nature paper.
Okay, the final bit of this analysis, and I'm thanking Jason for letting me use UCLA supercomputers soon. So here, what I'm trying to do.
I'm gonna process it.
Oh, you're gonna do it.
Yeah.
Okay.
So here what I'm doing is I want to, because so far, all of these things, I'm looking at them separately. But my point was to bring all of them together. So all of these, different factors. So I'm using a model that is called dominance analysis, that it's a glorified multiple regression. What it's doing, and this is why it's computationally heavy, it's trying to find all possible combination of all models of all predictors in predicting a Y. So more clearly, let's say our Y in our case is the area of the Hall, and we have 30 million different predictors, because we do. We have, cyto, we have micron, we have metabolism, and we have temporal properties, and we have receptors. Oh, you did?
Oh,
Okay, cool. So we have a bunch of other different things, but we know that the brain does not work in silos, right? So all of these things are related to one another. So what we want to do is to look at the individual or the relative contribution of each predictor when we account for its relevance compared to, for instance, let's say the serotonin receptor A, and how is serotonin receptor A predicting dynamics, but also receptor A in layer 1, or receptor A in layer 2, or receptor, the serotonin receptor and the GABA receptor. So it's all possible combinations. So it's a bunch of different models that we're running at once to try and, compare the incremental R squared across all sub models. That's it. Thank you, Andrew. Exactly. And all lower level models. So what I did so far to try and make it tractable is doing a PCA beforehand, before I throw in the dominance analysis. So this is just some, preliminary results before I send all my data to Jason so that we can run the full model. But here, what's really interesting is, again, replication between dynamics at the regional level and network level. So here, what we're seeing is that dynamics are shaped predominantly by cyto and micro, then followed by neuromodulation. So this is receptors, and then hierarchy. And it's not surprising that hierarchy is more contributing to functional connectivity because it's derived from functional connectivity data than RMSSD. But what I found very surprising is that it looks like more hardwired things, such as cyto and micro, are really shaping, the dynamics. Exactly. So yeah, so now the next step is to give all this breath. Thanks to Jason. So you're just gonna have to run one line of code for me. But basically, after we get that, what we can say is we can pinpoint what in the cyto domain is driving this, what in the receptor domain is driving it, instead of just being cyto overall. Okay, so importance of cyto and micro and shaping brain dynamics, followed by receptor density, replication among different data sets, and the importance of creating a comprehensive model as different aspects of brain structure and function are interrelated. Now, the last bit is just looking at age of cognition, and then I'm done, I promise. So here, basically, not surprising, what we're showing is that age is impacting dynamics and that there's lower dynamics at the SSD level, so the variability level. But what is cool with co-status is that you can really see how network flexibility or region flexibility is impacted, because as you can see, as we go from 20 to 86, the area of the hall is decreasing, right? So this is very remarkable, because this means that the dynamics of each region are shrinking. And so those regions are showing more stable dynamics. And what's interesting is that lowered brain dynamics are associated with age, but they also relate to stable trait measures of behavior, in that decreased brain dynamics are also associated with greater positive emotion and decreased negative emotion. And these are very preliminary results, but they may feed into the positivity bias that we know with older age. But again, I need to run more models on this bit. So, okay, taken together today, I hope I didn't bore you with all of these things. It was a lot, but I just hope that I'm driving home the point that we should care about brain dynamics, because our brain is intrinsically variable and dynamic, and all of these dynamic bits that I showed you overlap on different spatial scales, so regional and network, and they follow a precise topography. Brain dynamics do allow us to comprehensively delineate and characterize the functional brain organization, and are best predicted by cyto and micro factors along with neuromodulatory influences. And Max Shine will be happy to hear this. And that they decrease the function of age and are relevant for behavior. And then in putting on my methods hat again, we really need to make sure that we know that characterizing brain dynamics depends on the time scale over which these or said dynamics are measured. And that we should really consider a bunch of different, our tomato strain chain when we look at full signal variability. And it's really important to be transparent and report all of these methodological details from the moment we collect our data to how we establish variability, because then this will help the field try and validate and generalize the findings beyond just, one research group. And lastly, I hope you guys are as excited as I am about co-status, because I think it's cool. With that, I really would like to thank you all for listening to me, my lab, my undergrad EGU, my supervisor, and Jason and Lucina for collaborating with us, and all other collaborators at McGill, Toronto, and Verbanti at UT Dallas. Everyone.
This is really impressive. So what I want to do is if anyone on the Zoom wants to ask a question, we'll do that real quick, and then we'll go to a bunch and then people can ask you.
Absolutely.
So yeah, is there anyone who wants to ask anything before we let the Zoom crowd go? This is so much work. I mean, it's just really impressive. I hope you'll be able to write them up into several papers and not just one, because it's like too much for one paper. You could always follow up later. Julia's here all week, right?
Until Friday, yeah.
And we're hoping we'll get this variability grant, because then we can hire her as a postdoc. We'll all come together.
What's next?
After all this, let's say. And then we can hire Julia as a postdoc. So I put in a grant to look at variability in the ABCD data set, because there's like zero work on adolescent development of brain variability, and like zero work. So that's my hope, is that we'll just get funded to do these analyses forever and just keep doing them with ABCD and other data sets. That's what's I hope is next.
Yeah. Yeah, it is. Yeah, I'm just trying to wrap up, and I'm finalizing some analysis that I showed you today, and writing 3 million papers, I guess, out of these things, and then hopefully defend in a year and a half, 2 max.
Maybe my grants will come.
Exactly.
The time-
Apply for postdoc funding.
Yeah, we'll get all
That will
the,
help.
not all the stops.
Exactly.
Well, good. Well, let's go to lunch because we're hungry and thanks for joining us on Zoom.
Thank you. Bye-bye.
Thanks to Julia for